Hello everyone, my name is Natalia. I am going to tell you a little bit about my work, and specifically we're going to talk about the some of the aspects of lipidomic temperature adaptation in Mycoides and Syn. So I'd like to start with a little bit of general introduction. I am not sure that my slides are changing.
Not yet.
Not yet. My laptop is quite slow. I hope it works out.
If you need, you can send me your slides.
Okay. Does it work now? Yeah, hopefully. So we know that lipid membranes are essential to life. All living organisms, from single-cell bacteria to the most complex mammals, all have membranes around their cells. And we also know that throughout time, lipid membranes evolved to be these super complicated structures with very diverse genomes, with lipids of different kinds, and with a lot of membrane proteins associated with them. So we know that these lipidomes are very complex, and this is essentially one of the biggest experimental challenges of working with membranes. Even relatively simple experimental membrane models, such as E. coli or Bacillus, have at least 100 different lipid species in their membranes, and for mammalian cells, this number can be up to several hundreds of lipid species. On top of that, most of the common membrane model organisms, they have multiple membranes, which usually require quite some work to be separated experimentally in the lab, and they are also regulated by very complex transcriptional networks, which again are very hard to control. So essentially, in our lab, one of the first questions that we asked, if there isn't any alternative membrane model that would allow us to study the complexity and design principles of living membranes. Ideally, such a membrane model would approach the simplicity of a non-responsive synthetic lipid vesicle, yet possess all the functionality and adaptivity of a living membrane. And we found such a model in mycoplasmas, which we think are very exciting membrane model systems, because first off, they only have one membrane, so you do not need to go into elaborate separation experiments. They have a pretty limited lipid synthesis, so on average, they offer a somewhat simpler membranes than other common membrane model organisms. And what's very interesting about these bacteria is that they, as mammalian and avian parasites, they evolved to require sterols for growth, which is a very uncommon feature in bacterial world, and makes membranes of mycoplasma similar somewhat to the mammalian membranes. Thus, we also think that in the long term, mycoplasmas can work as a simple membrane models for very complicated, intricate mammalian plasma membranes. So essentially, for my project, we utilized Mycoplasma mycoides and Syn to investigate if mycoplasma adapts its membranes to environmental perturbations. Since these organisms are very simple, it was our very first question if they actually do adapt their membranes to change, say, in the growth temperatures. If they do, how does it happen? What are the mechanisms, lipidomic mechanisms behind their membrane adaptation? And in the long term, the big question is, what can we learn from mycoplasma membrane lipidome adaptations to temperature changes that would help us understand the fundamental principles of living membranes, design principles, and environmental adaptation? Essentially, the first thing we did, we looked at how mycoplasma lipidomes look like. And what we found out is that at least half of the entire lipidome is taken up by the exogenously picked up cholesterol, the only sterol in mycoplasma membranes, which is followed by major phospholipid classes, usually also present in mammalian and bacterial cells, such as phosphatidylcholine, PC, phosphodieldglycerol PG, cardiolipin, which is essentially double PG, and sphingomyelin, which is the only representative of sphingophospholipid in mycoplasma membranes. And as we can see, despite some differences in mole percent abundances of these lipid classes in mycoides and minimal cell membranes, in general, their overall lipidome structure is rather similar. So this picture essentially tells us that even lipidomes of such simple bacteria like mycoplasma are pretty much complex. So if we start at the most general characterization of lipidome components, mycoplasmas have two categories of lipids. First is sterols, represented by cholesterol, and another one is phospholipids. Phospholipids themselves in mycoplasma lipidomes are represented by glycerophospholipids and by sphingomyelin, which is the only sphingophospholipid in mycoplasmas. Finally, on the most detailed structural level, each of the phospholipids has at least two acyl chains characterized by length and unsaturation parameters, namely the number of carbons in the chain, which is length, and number of double bonds, which is the unsaturation. So it gives us quite a laboratory of possible lipid compositions in mycoplasma lipidome. So with those lipidomes, we asked ourselves, how do they, if they are capable of regulating these lipidomes, all of these components, and adapt their membrane properties in response to temperature perturbations. What I show you today is a particular example of how these bacteria maintain their membrane viscosity when they are grown at two different growth temperatures. One is 37 and another one is lower 30 degrees temperatures. So we measured the membrane viscosity of both mycoides and the minimal cell at these two temperatures and looked at how this viscosity changes with temperature. So if we plot these temperatures, if we plot the relative changes of membrane viscosity temperatures, what we see is that for mycoides, there is pretty much no difference. At both 37 and 30 degrees, the membrane viscosity remains pretty much constant, while for Syn3b, it's quite a significant change, which is several folds higher than for mycoides. According to the theory of homoviscous adaptation, if we do not observe any changes in membrane viscosity in the organism as it moves in a lower temperature, then we can say that this organism adapts its membrane property viscosity at different growth temperatures and maintains it very tightly controlled in some defined range. So for mycoides, it's pretty much the case, while Syn3b seemingly does a worse job in maintaining its membrane viscosities in a narrow range at lower temperature. So seeing this result, we asked ourselves, what are the lipidomic components that stay behind this observation? What is it that makes Syn3b adapt a little worse than mycoides does at 30 degrees? So if we come back to the phospholipid structural features pyramid and specifically focus on phospholipids today, then just as a reminder, in mycoplasma, there are glycerophospholipids and sphingophospholipid, which are structurally distinct. And for simplicity of this talk, we only going to focus on acyl chain features of these two phospholipid groups. So acyl chain features, again, are characterized by length and unsaturation of acyl chains in each of those phospholipid classes. So if we first start with unsaturation, namely the number of double bonds per acyl chain, and we talk about the average acyl chain features, meaning that I am not showing you the entire detailed distribution of double bonds and chain length in these phospholipids, but I'm rather calculating an average unsaturation and length per acyl chain and show you the aggregate number. Then what we can see if we calculate average unsaturation for sphingomyelin in Mycoplasma, Mycoides, and Syn3B, we can see that there is a very, very small change of this parameter in the minimal cell at lower temperature. No more than 1%, maybe even less than that. But it is somewhat larger for Mycoides. If we consider the same comparison of unsaturation in the minimal cell and Mycoides for glycerophospholipids, the picture is the opposite. Mycoides still does adapt its glycerophospholipid unsaturation by somewhat 5% at lower temperature, but for Syn3, this number is much, much larger. So this picture, this result shows us that it seems like while Mycoides implements a slight change of both sphingolipid and glycerophospholipid acyl chain unsaturation, for Syn3B, large modifications in sphingomyelin are pretty much not available, and instead the minimal cell chooses to modify the glycerophospholipids. Is it the same for the acyl chain length? So to show just the percentage comparison of how length changes at lower temperature for both Mycoides and Syn3, we can say that there is the same trend. While in general length is more conserved and less modified than the unsaturation in both glycerophospholipids and sphingomyelin, we can again see that for sphingomyelin, still Mycoides undergoes some shortening of acyl chain length in lower temperature, while Syn3B pretty much stays constant. And for glycerophospholipids, Syn3B tries to adjust the acyl chain length a little bit more than Mycoides does. We can also show the same result for sphingomyelin features if we plot the sphingomyelin average length versus its average saturation for Mycoides and Syn3B at both temperatures 37 and 30 degrees. As you can see, for Mycoides, there is a co-regulation of average features. The length goes down together with unsaturation at lower temperature, while minimal cell pretty much doesn't do much. And what this result suggests that it might be one of those specific mechanisms, lipidomic mechanisms, that are somehow missing or modified in the minimal cell that ultimately lead to worse homoviscous adaptation at lower temperatures. And though it's only one small part of big and complex lipidome of mycoplasmas, it is a very interesting observation of how a particular phospholipid class can be an active instrument in adaptation of membrane properties to environmental perturbations, which can affect the way membrane properties are maintained if it's not available anymore or modified somehow. And with this, I'd like to say that the future outlook for this is, of course, to identify what are the specific components in the membrane that lead to the alteration of this lipidomic adjustment mechanism between Mycoides and Syn3B, and that can explain more fundamental differences in how the membranes are constructed and adapted to different environmental perturbations. And with this, I'd like to thank my group and you for your attention.
Your group's, James's group's lipid stuff is just dazzling for me, Nataliya. So I would make the observation that while we minimized Mycoplasma mycoides to be, so we got rid of the genes that were not necessary for growth at 37. Wild type Mycoplasma mycoides has to be able to infect the upper respiratory system of a goat initially, which would be probably 30 degrees, and then the lower respiratory system of a goat later on, which would probably be 39 degrees. And so it makes sense to me that we have eliminated some of the capacity of the organism to live at the lower temperature.
Yeah.
Other questions for Nataliya? What's your next step, Nataliya? Go ahead. Shige.
Hello, I'm Shige from Japan. Nice to see you, John. And then again, John. And I have a question about the low temperature, the membrane viscosity. So at first, my question is, how did you measure the viscosity of membrane? And then we successfully obtained the low temperature of adapted strain of Syn3B. And what is expected to be happened in the membrane viscosity of the adapted strain?
Okay, good questions. So we measured the membrane viscosity in vivo, meaning that we did it in live cells, and we used the membrane dye, which is a derivative of C-lardin. And it's just essentially a hydrophobic probe, which is designed to sit in a hydrophobic environment in the membrane, and its fluorescence at different positions essentially tells you how lipid order is affected in a particular hydrophobic environment. So very strictly speaking, it's a dye that measures the lipid order of the membrane. So it gives you the general, we calculate the polarization number, which is just the relative ratio of different fluorescent channels, which is a readout for how ordered the membrane is. And it's a proxy for measuring the membrane viscosity in vivo. And to answer your second question, the cells were adapted at lower temperature. So all of the measurements, and both in vivo measurements and lipidomic measurements, are performed in the cells that are adapted to grow at whatever temperature for at least 3 consecutive passages. So what you see is it's not a transition stage. It's an adapted membrane. In I think if your question for how viscosity in general changes, so if you look at a lipid vesicle, which is non-responsive, just a lipid vesicle, then at the lower temperature, you would expect its lipid viscosity to go up, other things equal, because of the temperature. And essentially the concept of homoviscous adaptation in live organisms is that live organisms try to keep their viscosity constant to support the bioactivity of the membrane by inducing the lipidomic changes in it. And and it's a pretty well described concept observed in other membrane models as well, which suggests there's an active regulation of membrane properties when there is a challenge of lower growth temperature.
Okay, thank you very much.
Thank you, Nataliya.